Thank you, Professor Zaki, for your kind introduction, and thanks for the organizing committee for allowing me to share knowledge about this new topic. Okay, uh, the lecture, please. We will talk about diabetic kidney disease, and diabetic kidney disease, we all know that it is increasing, and I think now diabetes is widely present and it is expanding. It's known as epidemic, and we will talk about the role of autophagy on diabetic kidney disease, which is a very, I think there is a new topic. I will talk about the prevalence of diabetes worldwide, natural history, pathogenesis, and role of autophagy in diabetic kidney disease, evidence of the presence of autophagy in diabetic kidney disease, and then we will go to the practice and clinical implication. First, in IDF in 2021, emerged this data. More than 5 million adults live with diabetes worldwide in 2021. This will increase into 600 million patients in 2030. And this also will increase to more than 780 million people by 2045. 3 in 4 diabetic patients live in low and middle economic area. Diabetes is responsible for high mortality, 1 every 5 seconds mortality from diabetes, and it causes a big burden for the health insurance. And now we have another burden, which is the pre-diabetic, which will go into diabetic. More than 5 million adult has impaired glucose tolerance. In Hari, I think this is not new, but just we know that we have a progression of natural history of diabetic kidney disease coming from the increased kidney size and hyperfiltration, and then the presence of histological changes, then the increase in GFR, then normalized GFR, then decreased GFR to end-stage renal disease. During this stage, we have microalbuminuria and macroalbuminuria. And during the start and onset of microalbuminuria, I think hypertension start for this patient and then go to multiple risk factor to end-stage renal disease. I think the problem is too much, and we have already the patient with end-stage renal disease, 44% of them worldwide has or due to diabetes, but in Egypt we have less, so only 17% of patients in end-stage have diabetes. Diabetic kidney disease, or diabetic kidney disease, we considered an umbrella which include diabetic nephropathy and non-diabetic renal disease and mixed form and comorbidities like hypertension, obesity, and other problems. The pathophysiology or pathogenesis, briefly we call that we have a hemodynamic and metabolic factor in a genetically predisposed patient can lead to development of diabetic kidney disease. And I think now there is another area which is the acquired risk factor. Acquired risk factor, there is epigenetics modification, environmental changes, lifestyle, oxidative stress, hypoxia, and chronic inflammation. All this will contribute to diabetic kidney disease. And here hyperglycemia through some of cytokines and growth factors do epigenetics with microRNA, and this will lead to the pathophysiology and the pathology in the kidney. And we have already inflammation. Then with inflammation, I have apoptosis, and I have already delayed or decreased the autophagy in this patient. This will lead to hypertrophy and finally fibrosis and reaching end-stage renal disease. So I think now this is another concept. This chronic hyperglycemia lead to altered autophagy activity, and this will affect all cells in the kidney. The podocyte, by podocyte damage and hypertrophy and apoptosis, then later on decrease the filtration and microalbuminuria, kidney fibrosis, inflammation, already glomerulosclerosis, tubular cell, podocytes, and mesangial cell, all cells are be affected, and they put the altered autophagy as one of the pathogenesis for these pathways. We need to tell what is the autophagy. Autophagy in Latin means self-eating. This means that I will take the abnormal organelle in the cell and try to make it removed from the cell. So this is healthy, and this is help for recycling of the cell and make the cell survive more. So everything which can stimulate autophagy will make me more healthy, and everything will inhibit autophagy will make me more disease. Just we'll take this is a cellular mechanism that transport the intracytoplasmic component to lysozyme to preserve cellular function and homeostasis. Autophagy integrity is essential for cell homeostasis. Its alteration can drive the cell to damage and death. Defective autophagy signal is now found in multiple diseases: autoimmune disease, infectious disease, metabolic disease, type 2 diabetes, diabetic kidney disease, cardiovascular and pulmonary disease. Autophagy controlled by gene, and mutation in these genes will cause also disease. Now there is intense research ongoing about explaining the drugs which is working in diabetes and diabetic kidney disease by autophagy mechanism. Again, we need to make isolation of the from one of the membrane. We have some. Okay. Okay. There is initiation with the presence of membrane, and this membrane cause some of the diseased organelle. Okay. This membrane, this called phagophore, and this case takes the diseased organelle in the cell and then become elongated, then maturated, then combined or fused with lysozyme to make it autophagolysozyme. Then breakdown and degradation occur for the abnormal organelle. And this is healthy because they use these amino acids and nucleic acid to build up the cell again. Nobel Prize will be awarded to a Japanese scientist named Yoshino Osimi, and this in 2016 because he discovered the mechanism of autophagy. We will go to some molecular basis of this autophagy, and I have two important signal. The first AMB kinase signal, which is an activator of the autophagy, and emitter signal, which is an inhibitor of autophagy. One is important molecule. This is ULK1, and this when autophagy stimulated, it's increase. And this will lead to multiple molecule, and think multiple molecule, more than 100 molecule may be associated with occurrence or of autophagy. And this, what is important is microtubule associated protein, which is LC3. This is very important, and autophagy related genes. I think this all is stimulated on the molecular basis, and secretion of a lot of things help to stimulation of autophagy, and this will help the cell to survive. I think secretion, the way of secretion of this protein, either حاجة what's called conventional أو canonical، يعني جاية عن طريق endoplasmic reticulum of Golgi apparatus. Sometimes it's not go in this traditional pathway and called it non-canonical, and this will lead to excretion of this protein without reaching the endoplasmic reticulum or Golgi, and this is named secretory autophagy. In kidney, as we all know that now, it's helped to preserve the kidney function. Autophagy, reflux or flux, critical in maintaining renal podocyte, proximal convolute tubule, epithelial cell, mesangial cell, endothelial cell, and contributed to normal renal homeostasis. In diabetic kidney disease, there is increasing evidence shows that defective autophagy in diabetic kidney occur. Autophagy deficiency in diabetic kidney may increase the susceptibility of the kidney cell to diabetes-associated damage. which in turn lead to treatment-resistant albuminuria and progressive decline in the kidney function. Restoration of autophagy activity may become a new therapeutic option to protect the kidney. As we know, as podocyte injury occur early in patients with diabetes, and most of diabetic there is found podocyte-derived parts present in the urine in early stages of diabetes, and we know that the podocyte has no capacity to regenerate, and when this undergo apoptosis and death, so fibrosis. So decrease the amount will affect the function, and this lead to affection of the glomerular basement membrane and albuminuria and progression to end-stage renal disease. So here may be more clear. Starvation, one of the stimulant of autophagy by inhibition of mTOR, and also by inhibition of ATP and increase the activity of AMP kinase. This will help to stimulation of autophagy. Endoplasmic reticulum stress also lead to activation of autophagy. Activation of autophagy lead to inhibition of podocyte apoptosis, food process effacement, and CKD progression. Also, now we have mTOR and we have rapamycin. Inhibition by rapamycin will activate autophagy, and this will help to improve the kidney. If we do the reverse, mTOR activation results in podocyte damage because it inhibits autophagy. And mTOR by rapamycin will increase the marker that denotes that autophagy is working, like LC3, and this makes more protection to the podocyte. As regards proximal convoluted tubules, already we have some of anything which can stimulate autophagy will inhibit tubular cell damage and kidney fibrosis and deterioration of the kidney function. But we have here chromic hypoglycemia. This will inhibit autophagy. Deletion of the autophagy gene will inhibit autophagy. AGIB and with its receptor will inhibit the autophagy, and this will lead to more destruction of the kidney. This is another evidence for this because ULC1, I think this is the product, first product for autophagy. So if down regulation of this substance, this means that autophagy is inhibited. And they found that it is inhibited by upregulation of some of the microRNA. And another thing, they make ablation of this microRNA. They found that autophagy is improved and this marker is increased. Another marker for autophagy, here we found when stimulation of autophagy, there is some protein like P62. This protein will decrease by autophagy. So if I found it high, this means that autophagy is suppressed. And this is indicator of more progression of the kidney disease and destruction. We have only two markers previously or now for measuring the presence of diabetic kidney disease, which is estimated GFR and albuminuria. And I think this is not enough tailored therapy for this. The new is the molecular multi-marker panel sensor, which is show dysregulation of various problems and various pathogenic mechanisms. I think this will be more helpful. So we will go another parameter, another approach by genomic, transcriptomic, proteomic, metabolomic, and this is need, I think, data system collection and bioinformatics, and this may be change for the era. Is this has an implication for the therapy? Yes, they found the search in anti-diabetic drugs, which is effectively used in diabetic kidney disease, and they found that some work by AMP kinase activator like metformin, SGLT inhibitor, PPAR alpha and gamma, glucagon, GLP-1 agonist, all these will stimulate autophagy. Okay? mTOR inhibitors, also we know that inhibition of mTOR will lead to stimulation of autophagy, like rapamycin, everolimus, and MBK activator. Some others which is not dependent on AMP or mTOR زي براxidolone. This is an activation of antioxidant ليها pathway عن طريق nuclear factor erythroid 2 related factor and apoptosis signal regulating kinase and some drug like with anti-inflammatory activity. First, metformin. Metformin is first line treatment in diabetic patient all, and all guidelines recommend the use it till contraindication, and he found that it stimulates AMP kinase dependent and also acts through mTOR by AMP independent pathway, and found that it increased the insulin sensitivity, gluconeogenesis, improve GLP-1 secretions and microbiota and redox state. This is another for metformin and autophagy and podocyte protection. SGLT, this is working by the two, by AMP activator and mTOR suppression single, and have a lot of mediators. So it's associated with glucagon, it's associated with hypoxia-inducible factor, EPO concentration, hemodynamic effect, change in glomerulotubular balance, RAS and sympathetic tone inhibition, and has already proven cardiovascular and renoprotective effect. And this is, I think, said we see today that this is a beneficial for all patients by all study for renoprotection. And this is the mechanism of action. And now I don't go to all through, but we have here cardiovascular, hemodynamic, tubuloglomerular feedback, proximal convoluted tubule hypertrophy, podocyte, and here reactive oxygen species and AMPK activator. So it's proof that it works and stimulates autophagy. Another work by SGLT through glucagon. What's occurring in glucagon? This is normal person. He has already in the morning biogenesis and a night mitophagy. This is some sort of autophagy occurring during fasting, and this keeps the cell healthy. By using insulin, we have biogenesis but very low mitophagy. But when we insert the SGLT to this patient, again, biogenesis alternating with mitophagy to normal, so it helps to stimulate autophagy through glucagon. GLP-1 receptor is mediated action خلال AMP activator dependent and also independent about another thing like natriuresis and NH3. GLP-1 agonist, we have exenatide, liraglutide, dulaglutide, semaglutide, all are highly effective anti-diabetic agent and have renoprotective effect which mediated by GLP-1. This is the effect for decreased inflammation, oxidative stress, podocyte loss, also decreased tubular injury, decrease renal fibrosis and glomerulosclerosis, and preserve renoprotection and cardiovascular protection. But actually, the role of autophagy as potential explanation of this benefit still uncertain. RAS blockade, we all use it in patients with diabetic kidney disease, and it's recommended to use it for high dose. It also has work about AMP kinase and mTOR mediated clinical proved benefit, but actually the role of autophagy as a potential explanation for its benefit is not certain. Antifibrotic with new mineralocorticoid inhibitors, fenfrinone, I think they have inhibition pro-inflammation will pro-fibrosis, and now I think a lot of study for this, but maybe the role of autophagy is not certain. Rapamycin, erythromycin, is the other mTOR inhibitors. It causes direct inhibition in mTOR, and this will activate the autophagy. But we use it. I don't know. It's not use it, but it's seen in some models in biopsy of human transplant patients. Some independent of AMP or mTOR, like bardoxolone, the activator antioxidant, act على nuclear factor erythroid 2 related factor, apoptosis signal regulating kinase, Some anti-inflammatories, these are a lot, but maybe bentexifylline, this is the Trental. Inhibition بيعمل inhibition للcell proliferation والkidney inflammation والaccumulation of extracellular matrix and use through autophagy also inhibition. This is Walt Disney, the way to get started is to quit talking and begin doing by making a new era, making a novel biomarker for diabetic kidney disease. And I think this might be last slide. And this diabetic kidney disease is the most medical and economic burden affecting the diabetic patient. Impaired autophagy involved in pathogenesis of diabetic kidney disease. I just okay. I can conclude the other. Okay, we found that autophagy disturbance involved. It is present in pathogenesis of diabetic kidney disease, glomeruli, tubule, interstitial cell, and vascular renal compartments suffer from impact of autophagy dysregulation in different stages of CKD. Understanding this underlying molecular mechanism involved in pathogenesis will changing the face of diabetic kidney disease and resulting in more understanding and better application of the new tool for measurements. But actually, critical information modification of autophagy by potential nephroprotective agent is still lacking. And most of this study is animal study. So human model for inhibition of autophagy still not need more further research. And thank you.
Thank you, Dr. Eman, for your very informative lecture. But I want to ask a question. We repeat this question many times. To what level of GFR we can use metformin الدواء الساحر بتاعنا؟
What?
To what level of EGFR we can use metformin?
Yeah. I think metformin is present in all guidelines not to be used below 30. Below 30. If GFR below 30, better to stop it. He has renal protection and a lot of very good, but sometimes there is lactic acidosis which is associated with, despite it's very rare. Despite it's very rare. And some people, but it's not present as a guideline, use it off-label less than that by lower dose.
بشكر حضرتك الأستاذة الدكتورة إيمان، بس أنا ليا تعليق على سؤال أستاذي الفاضل. إحنا according لل ADA guidelines 2022 we all know that the effective dose of metformin is 2 جرام. في ال estimated GFR لو 45 هنعمل dose modification ل halving the dose 1 جرام. If ال estimated GFR 30 أو أقل don't initiate.
Yes, yes. This is the guideline, actually. Okay.
Okay, thank you for the lack of time.
Thank you.
End this session. Thank you for all the speakers and audience. Thank you.